DNA replication is important as it must occur for cell division and will occur in the nucleus for eukaryotic cells, but in the cytoplasm for prokaryotic cells. DNA replication will occur during cell division, specifically during the synthesis phase of interphase. DNA has a very specific structure, with this strand being known as the leading strand running 3' to 5', and this strand known as the lagging strand running from 5' to 3', which that's determined by the number of the carbon atom on the deoxyribose that a phosphate group covalently bonds to. The deoxyribose sugar and phosphate group are known as the backbone of DNA. And these two strands of DNA run anti-parallel. The phosphates, sugars, and nitrogen bases all have covalent bonds, which are represented by the blue strings, and the nitrogen bases to each other from opposite strands are hydrogen bonded, which are represented by the brown strings. The nitrogen bases we have are adenine, which is represented by a red pin, thymine, which is represented by a yellow pin, cytosine, which is represented by a blue pin, and guanine, which is represented by a purple pin. Now to start off with DNA replication, we have the helicase, whose job is to break the hydrogen bonds in between all the nitrogen bases in order to continue with DNA replication. Now that those bonds are broken, SSB proteins will attach to the nitrogen bases to make sure they don't reattach. These SSB proteins are represented by the tiny blue gems. Now, over here, we have topoisomerase, an enzyme that prevents supercoiling of DNA. Now, this is in order to make helicase's job easier to break those hydrogen bonds by breaking, swiveling, and rejoining parental DNA ahead of the replication fork, which was done by the helicase. From here, a primase is going to leave a primer on the nitrogen base right here, signaling DNA polymerase to start replicating the DNA. DNA polymerase 3 will come through here and bond all the nitrogen bases with the deoxyribose sugar and the phosphate group attached to it all the way throughout this DNA strand without stopping, because this is the leading strand. After polymerase 3 comes through, polymerase 1 also will come through to check for any mistakes made by DNA polymerase 3 that did not get addressed. And here we have our completed DNA segment for the leading strand. To sum up the DNA polymerase 1 and 3, DNA polymerase 1 fills the gaps in DNA formed from replication, repair, and recombination in order to make the DNA whole, while DNA polymerase 3 is the main it's the main enzyme that puts the nitrogen bases and connects to make the DNA whole. Now, finally, from the lagging strand, we have a primase once again, except this time it's going to be a little bit different because of the fact that this is the lagging strand. Now, because of this, the primase moves in. It has to move in a certain direction, going from the 3' to 5'. So it must move backwards from the way the helicase was moving across, which will be important in a few seconds. So the primase moves across, and it will put down primers.